Hi dear students, I am Dr. Mashur. Welcome to my class. Today I would like to discuss selection and screening of recombinant clones. Okay, in recombinant DNA technology, I would like to discuss selection and screening of recombinant clones. Let's see selection and screening of recombinant clones. Okay, basically this procedure of selection and screening of recombinant clones is performed to identify a clone of individual specified gene. Okay, so we have discussed in last classes that the different structures, different steps involved in gene cloning. So okay, so after transferring the recombinant DNA into the host cell, we'll get number of colonies. Okay, after transferring the recombinant DNA into the host cell, we'll get number of colonies of bacteria. That time, that some colonies only contain the specific DNA portion. Okay, specific recombinant DNA. Okay, here you can see we having some one gene is here, one DNA portion is here, large DNA molecule is here, then there also contain different gene is there, but this red gene is only our specified gene. Okay, red gene is the only specified gene. Okay, so that we have digested with one enzyme, so we'll get many fragments, and we have cloned all this fragment into the vectors, and we transform into bacteria. So that time we'll get number of colonies here, but we don't know which colony contain our specific DNA molecule. Okay, our specific DNA molecule. For that, we have to screen this. Okay, we have to screen this all cells to identify which colony containing our specific DNA molecule. Okay, or specific DNA molecule, which colony contain a specific molecule. For that purpose, we will do the screening of recombinant clones to identify which clone of bacteria or which cell contain the uh, exact desired DNA molecules. Okay, present in which colony. So this the selection and screening process of recombinant clone is used to identify the a specific clone that contain the individual specific gene. Okay, and this identification of this specific clone can be carried out by exploiting either by the DNA sequence of the clone or its structure. Okay, or we can adopt the function of its expressed products. Okay, we told that we have to identify the exact clone from this mixed clones. So for that, to identify that, we'll exploit its some properties of these clones. So we'll check the either sequence of the clone or the structure of that clone, or we'll take the functions of its that gene expressed product. That means functions of its expressed products means we'll check the protein, if the protein is present in that clone. Okay, for that identification of specific clone, we'll adopt either the DNA sequence of that clone, or we'll take the structure of that clone, or otherwise we'll take the functions of that gene or that expressed products. Expressed product means that gene coding a protein. Okay, and so that there are different procedures is there to identify desired clone. Okay, there are many different procedures is there by which the desired clone can be obtained. If one of there are different procedures, and these are the some of the major and commonly using procedure to identify the desired clone from the mixture of clones. Okay, then the first one is direct selection, and second one is blue-white screening, and third one is colony hybridization, and fourth one is plague hybridization, and fifth one is immunological method, and sixth one is PCR. Okay, we can see there are when we transfer the recombinant DNA in the host cell, that time in the bacteria in this agar medium will get number of colonies. Okay, we'll get number of colonies. That number of colonies, we have from that number of colonies we have to identify which is our desired recombinant DNA molecules. Okay, which is our desired recombinant molecule we have to identify. For that procedure, we'll screen the recombinant clones, and for this screening procedure, will exploit either it's the DNA sequence of that specified DNA or the structure of that clones, or will exploit the functions of that genes expressed product. Okay, these are the method we'll adapt to, will exploit to identify the desired clones. Okay, and there are different procedures by which they are used to identify the desired clones. And the first method is a direct selection, second one is a blue-white screen, third one is a colony hybridization, and fourth one is a plague hybridization, and fifth one is a immunological method, and sixth one is a PCR. So we can see the next we can see the individual method separately. So we have told that there are different techniques used for selection of clones. Okay, and this is the first technique, that is a direct selection of recombinants. Okay, direct selection of recombinants. Okay, in this procedure the desired clone only will be survive on the selection medium. Okay, in this procedure the desired clone only will be that means the required the clone of required gene only will be survive on the selection medium. Okay, to select the clone. Okay, to select for a cloned gene, it is necessary to place the transformants onto an agar medium on which the only desired recombinant can grow and others cannot grow. Okay, to able to select this for a cloned gene, we have to place the transformants on an agar medium. On that medium the desired clone can only able to grow and others cannot able to grow. Okay, and we can discuss this method by using a simple example. Okay, we can explain this method by using a simple example. So here you have to clone a antibiotic resistant gene. Okay, in this procedure we can amplify a specific antibiotic resistant gene into the another bacteria. We have to clone an antibiotic resistant gene on another bacterium. Here you can see one plasmid is there. Here you can see one plasmid is there. Okay, one plasmid, his name is R65 plasmid is there. R65 plasmid is there. Okay, R65 plasmid is there, and that contain one gene, kanamycin resistant gene is there. Okay, kanamycin resistant gene is there, and this plasmid also contains some other antibiotic gene like kanamycin, chloramphenicol, streptomycin, and sulfonamide also present in this plasmid. But we have to clone this kanamycin gene only. Okay, we have to clone this kanamycin gene only. Okay, and we can see here, you see this kanamycin gene is present inside a EcoR1 site. Okay, this lines indicates the EcoR1 site. Okay, this line indicates the EcoR1 site. So we cut this plasmid. Okay, we have to isolate we have to clone this kanamycin gene in the bacterium. So we'll cut this plasmid using EcoR1. Okay, we'll cut this plasmid using EcoR1. So that time we'll get around 13 different fragments. Okay, we'll get a 13 different fragments. Okay, but in that our desired fragment, kanamycin gene fragment, is present here, and that will clone. Okay, we cannot able to isolate this individual fragment from this mixture of fragments, so we have to clone this total 13 DNA fragments into the echo R1 site of PBR322. Okay, echo R1 site of PBR322, and we'll insert into the plasmid PBR322. Okay, here you will get around that 13 fragments transform that recombinant DNA here, and we'll transfer that recombinant all these 13 fragments into the bacteria. Okay, we'll transfer the E. coli bacteria, and after that we'll get, okay, we'll transfer this E. coli bacteria, and we'll grow that transformed E. coli bacteria in kanamycin containing agar medium. Okay, we'll transfer this that transformed E. coli bacteria into the we'll grow the transformed E. coli bacteria in the kanamycin containing medium. Okay, kanamycin containing medium. Okay, that bacteria, host bacteria, is susceptible to kanamycin, and this medium contain kanamycin. Okay, that time we'll get only one colony. Okay, we'll get only one colony. That colony contain kanamycin resistant gene. That colony all contain kanamycin resistant gene, because we told that this medium contain the kanamycin. So other dual recombinants does not have the kanamycin gene, so that transformed bacteria cannot able to survive either kanamycin contain, but this bacteria contain the this fragment kanamycin, so that colony is will be survived here. So we can directly select this colony from here. That's why it is called as a direct selection. Okay, this is the method of direct selection of recombinants. Okay, in the process of direct selection of recombinant DNA, that time the desired gene containing clone only will be survive in the selection medium. Okay, the medium that desired gene containing clone only will be survey. For that we have to plate the transformers on the selective medium, and that medium only allows that desired DNA product to grow on that medium. Others cannot able to survive on that. For that we can explain this method by using a simple experiment. Okay, we can explain this that method by using a simple experiment. Here one that we can explain that by using a cloning of a antibiotic resistant gene cloning. Okay, here you can see one plasmid is there, R65 plasmid is there, that's contain one gene kanamycin, and that kanamycin is present inside the echo R1 fragment. Okay, echo R1 fragment. Here echo R1 site is there, echo R1. So when we cut here, they'll form a kanamycin free kanamycin fragment. But and this plasmid also contains some other antibiotic also is the chloramphenicol, streptomycin, sulfonamide is there. But we have to amplify this kanamycin. We have to clone this kanamycin. For that we'll cut this plasmid with the echo R1. Okay, we'll cut with the echo R1. So we'll get a around 13 fragment we'll get. In that one fragment will be the fragment of kanamycin resistant gene. Okay, kanamycin resistant gene. So we'll clone this, we'll incorporate this fragment into the PBR322. Okay, we'll incorporate this fragment into PBR322 and transfer into the bacteria. Okay, transfer into bacteria, and that transformed bacteria will be allowed to grow in medium containing kanamycin. Okay, transformed bacteria is allowed to grow on the medium containing kanamycin, but only one clone will be survive here, because that only bacteria containing kanamycin resistant will be survive here. Okay, only the bacteria that contain kanamycin resistant gene only will be survive here, because other that bacteria is a kanamycin susceptible, and that this recombinant also does not contain kanamycin. So that's this unwanted, this kanamycin absent bacteria cannot able to survive here. So we can directly select the recombinant kanamycin clones from here. This is the direct selection of recombinants. Okay. And the next method is a blue-white screening method. Okay, blue-white screening method. This method is widely used for screening of to get recombinant clones. Okay, this method is widely used for screening of recombinant clones. Okay, in this gene cloning procedure, there is a one vector is there. That vector containing a selectable marker gene, laxid gene, and an antibiotic resistant gene, ampicillin resistant gene. Okay, this gene cloning method use a vector called that vector containing a selectable marker, a laxid gene, and an antibiotic resistant gene, ampicillin resistant gene. Okay, that laxid gene encodes an enzyme called beta galactosylase. Okay, that laxid gene encode a protein called an enzyme called beta galactosylase. At that beta galactosylase cleave the substrate S-gal in the S-gal present in the medium to blue colored insoluble product. Okay, when we transfer this laxid gene into the bacteria, that time that bacteria will produce blue colored colony. Okay, that bacteria will produce blue colored colony due to the presence of this laxid gene. Okay, this laxid gene coding enzyme beta galactosylase cleave the colorless substrate S-gal which is present in the medium to blue colored insoluble product. Okay, this beta galactosylase cleave the substrate S-gal present in the medium to the blue colored insoluble product. Okay, when we insert a foreign DNA into the laxid gene. Okay, when we insert a foreign DNA into the laxid gene, that laxid gene will be inactivated. Okay, that laxid gene will be inactivated. So that process is known as a insertion inactivation. Okay, when we insert a foreign DNA into the laxid gene, that time that laxid gene will be inactivated. So that inactivation process is known as a insertion inactivation. Okay, once this laxid gene is inactivated, that time this laxid gene containing colony cannot able to produce blue colored colony. Okay, once this inactivated laxid gene present in the bacteria that cannot able to produce blue colored colony. Okay, this laxid inactivated laxid gene only produce white colored colony. Okay, insert foreign DNA into this, okay, that laxid gene will be inactivated. Here you can see this is the a vector. Okay, this is the vector that containing the laxid gene here, and here ampicillin resistant gene. Okay, when we this is a foreign DNA, we'll insert the foreign DNA into the laxid gene. Okay, we'll insert the vector laxid gene will produce, we'll insert the laxid gene into the laxid gene of the vector. Okay, then after inserting this this foreign DNA into the laxid gene, we'll transfer this recombinant vector into the host cell. Okay, recombinant vector into the host cell, and so that time there's chances that we'll get some colonies. Here you can see this foreign DNA is correctly inserted into the laxid gene of this vector. Okay, this foreign DNA is correctly inserted in the foreign laxid gene of this vector. So this bacteria will produce, this laxid gene is absent, so inactivated. So laxid gene is inactivated. That means this is known as insertion inactivation. Okay, this laxid gene is inactivated, so this process is known as a insertion inactivation. And this bacteria laxid gene inactivated, that's why it's produced only white colored colony. Okay, only white colored colony, because this bacteria can able to survive, and this medium, this all medium will be ampicillin containing medium. Okay, ampicillin containing medium. Okay, this bacteria will be, this medium should be ampicillin containing medium, and this the vector containing foreign DNA in the laxid gene, that will produce blue colored colony in the ampicillin containing medium. Okay, but because it can able to survive in the ampicillin medium, because it having a fluorescent gene of ampicillin is here, so I can produce white colored colony. Okay, in this another one type of bacteria will also use with in this mixer. That's having no insert in the laxagine. Okay, no insert in the laxagine. So that no insert in the laxagine. That's why laxagine will be active. Okay, laxagine will be active. So this bacteria, this bacteria having what? This bacteria having the vector, but the foreign DNA is not inserted in the laxagine. So that's why this bacteria, laxagine is activated. That's why it's produce blue colored colony in the ampicillin medium. Okay, that's why it's produce a blue colored colony in the ampicillin medium. Okay, sometime this foreign DNA will be inserted outside this, or inserted the outside the laxagine. That time also it will produce blue colored colony. Okay, sometime in this mixer some one bacteria also is there. That bacteria does not contain the vector, does not contain the vector. That bacteria cannot able to survive in the ampicillin containing medium. Okay, that's bacteria cannot survive in the ampicillin resistant medium. Okay, so here you can see this is a blue colored colony. This blue colored colony is because of this foreign DNA is not inserted in the laxagine. Okay, the foreign DNA is not inserted in the laxagine. If foreign DNA is inserted in the laxagine, that laxagine will be inactivated. So the inactivated laxagine, that means that contain the foreign DNA, so that produce the colorless white white colored colony. So this white is the recombinant clone. So, okay, this white is the recombinant clone because there's a laxagine inactivated due to the presence or due to the insertion of foreign DNA to it. Okay, so we can select this white colored colony as a recombinant clone, and this blue is because there is no insertion of foreign DNA in the laxagine. Okay, this is the blue white screening method. Okay, this is the blue white screening method. Okay, from this, on the basis of the basis of color, okay, the basis of color, we can identify the recombinant clone in this blue white screen. That means the white color colony means there is a recombinant clone, and the blue means there is a unrecombinant clone. That means blue does not contain the recombinant DNA, and white contain the recombinant DNA. Okay, this is the blue white screening. The next technique is a colony hybridization. Okay, for the screening of recombinant clone, the next technique is a colony hybridization. Okay, using this colony hybridization technique, we can rapidly determine which colony having the desired DNA product, and we can screen this DNA product from the thousands of colonies. Okay, that means the colony hybridization technique can rapidly determine which colony among the thousands contain the target sequence. Okay, from thousands of colony, we can identify which colony having our desired product using this colony hybridization technique. Okay, and in the steps of colony hybridization, okay, we can see what are steps involved in the DNA this colony hybridization technique. Okay, in that we having a plate of medium containing transformed colonies. Okay, plate of DNA containing the transformed plate. Okay, transformed bacterial colonies here. Okay, this transformed bacterial colony is here, and this colony hybridization mainly used in the case of to screen the genomic library. Okay, genomic library or cDNA library, we can use this colony hybridization because thousands of hundreds of colony will be there in this colony genomic or cDNA library preparation, so that from there we can easily isolate this our desired DNA containing colony using this colony hybridization. Okay, this is the plate. This plate is contain different colonies. Okay, colonies. We have to isolate our desired DNA containing colonies from this plate. Okay, we have to isolate desired DNA containing colonies from this plate. Okay, so that we'll keep this place as a master plate. Okay, we'll keep this uh, plate as a transformer plate is kept as a master plate. Okay, we'll kept as a master plate, and using this sterile velvet block, sterile velvet block, we'll make a replica. Okay, we'll using this part, we'll plus this here, and we'll transfer the colonies into, okay, the portion of trans colonies, DNA of these colonies will be transferred into nitrocellulose membrane placed in a petri plate. Okay, using this, this will be plus here this direction. Okay, this part will be plus in this direction, and will transfer, make a replica plate on nitrocellulose membrane placed in a petri plate. Okay, placed in a petri plate. Okay, so this colony, copy of colony is transferred into here. Okay, after that we'll replace this nitrocellulose membrane from here. We'll remove the nitrocellulose membrane from here, and we'll treat this plate. Okay, remember that this this membrane contain the copy of this colonies. Okay, copy of this colonies here, and after that we have to lyse this bacteria. Okay, lyse this bacteria using NaOH. Okay, using NaOH will lyse the bacteria. That time that NaOH will degrade the rupture the cell membrane of this bacteria, and also it will uh, denature the bacteria, denature the DNA of the bacteria, and that form a single standard DNA. Okay, single standard DNA, it will be here, and remaining DNA will be remaining DNA will be detached from here, and after that we'll treat with the proteinase enzyme to remove the proteins, and we'll after that we'll wash, and we'll bake this nitrocellulose membrane with a at a 80 degrees Celsius. At 80 degrees Celsius will this bake. That time that single standard DNA will be tightly bound with this nitrocellulose membrane. Okay, this the single standard DNA that we got after this NaOH treatment, that will bound tightly with this nitrocellulose membrane, and that binding is with a sugar phosphate backbone. Okay, the single stranded DNA will be bind with this nitrocellulose membrane by a sugar phosphate backbone, and that free bases will be ready to bind with this other complementary DNA. Okay, when the single stranded DNA is bound with this nitrocellulose membrane, that time the sugar phosphate backbone is will be attached into the, that helps to bond with the nitrocellulose membrane. That time that is bases will be free there. Okay, and bases will be free there, and after that now we got that this plate with only this having DNA bound over here. This is a DNA bound on this nitrocellulose membrane. Okay, after this, this is the these are the colonies that we treated with NaOH and protease, and we bake in 80 degrees Celsius. That, and after that we'll washing, we'll get a nitrocellulose membrane with only DNA. Okay, this is only DNA, and after that in this, thus, okay, this is the DNA print of this bacterial colonies. Okay, this is the DNA print of this bacterial colony, and into this nitrocellulose membrane we'll add a radio labeled probe. Okay, to this nitrocellulose membrane we'll add radio labeled probe. Okay, that radio labeled probe will be highly specific to our target DNA. we know that probes are labeled oligonucleotides. Okay, there are the probes. When we add the probes to this nitrocellulose membrane, that specific probe will be bind with its target DNA. Okay, when we add the probes to this nitrocellulose membrane, that time that probe will be bind with its specific target DNA. Okay, after that, after this adding of probe, we will allow to hybridize on that nitrocellulose membrane containing DNA, and after that we'll wash after an incubation, after a period of incubation, and at that time this unbound probes will be removed from this nitrocellulose membrane, and we'll get a bound probes on this. On that time we'll get only bound probes on this nitrocellulose membrane. Okay, and to check on which colony containing DNA this probe is bound, the time will expose this nitrocellulose membrane undergo for autoradiography, because these probes are radiolabeled. Okay, these probes used in this case that are radiolabeled, so we'll go for this nitrocellulose membrane undergo for autoradiography and will expose into X-ray film. That time we'll get a spot here. Okay, we'll get a spot here. That spot, that is a that's contain the our desired DNA. Okay, that's desired program DNA. So this spot will be compare with our master plate. Okay, we'll compare with the master plate. So here you can see the same location. Here you can see a colony. Okay, you can see the master plate. You can see the same colonies here. So we can say that this is the colony that containing our desired DNA. Okay, this is the colony contain our desired product. Okay, because this in this nitrocellulose membrane, this DNA, this colony's DNA is showed the radio autoradiography. So that that's given the radio signals. So we can say that this in the X-ray film, this spot we can identify, and this spot is corresponding location. Space we can compare here, compare here. So we can finalize that this is the colony is the having our desired recombinant DNA. That means these are the recombinant clone. Okay, this is the colony hybridization. I hope you understand this. Then the next method is the plaque hybridization technique. Okay, plaque hybridization technique. Okay, this plaque hybridization technique is similar to colony hybridization. Okay, is similar to colony hybridization. In colony hybridization, we'll hybridize the colonies, but here we'll use this hybridization of phages of this plaques. Okay, plaques will hybridize the plaques. I think you know what are the plaques. What are the? Yeah, I think you know what are the plaques. Okay, plaques are the clear zones, clear areas you can see in the bacterial plate when that will be infected with phages or that virus. That means viruses. Okay, plaques are the clear areas, zones or point that is found in the bacterial plate that will be infected with viruses. So you can see that you can say that plaques as a clear zone. Okay, here this plates, these are the clear zone. Here this is the bacterial plate. Here you can see that some plaques here. Here's this bacterial area that is infected with that viruses or bacteria phages. That's why it is destructed the bacteria, and this plaques will contain our this phages. So when in the recombinant DNA technology, when we are using phage vectors, okay, when we are using phage vector, we'll transfer this phages into the bacteria. Will play in the bacteria. That time that phage will infect the bacteria, and that phages will kill the bacteria, and that will provide, that will create some clear zones like this, clear zone like this. So these are the plaques here. These are the plaques. Okay, these are the plaques, and this plaques will contain different recombinant phages. Okay, this plaques will be created by different recombinant phages. So we have to isolate which plaques having our desired recombinant phage. Okay, we have to identify which plaques containing our desired recombinant phage. That means which phage containing our desired DNA. So that here you can see these are the plaques. Okay, these are the phage plaques. This one phage plaque and this one other phage plaque. Okay, these are the phage plaques. So for that we'll get the transformed phage plaques here. One plate will be here. So we'll keep this plate as a master plate. We will keep this as a master plate. I told that procedure is exactly similar to our colony hybridization. Okay, we'll keep this plates as a master plate, and we'll go for a replica plating of this plate, kind of plaques plate, into the nitrocellulose membrane. Okay, nitrocellulose membrane will transfer this plaque or a replica plating, and in this nitrocellulose plate we'll treat with NaOH. Okay, NaOH protease and we'll bake this nitrocellulose membrane with that's 80 at 80 degrees Celsius. Okay, that 80 degrees Celsius. So that time NaOH will cleave break this phage covering, and that will denature the double stranded DNA present in the phage and is converted into single strand DNA. That single stranded DNA will be tightly bound with this nitrocellulose membrane when it's baked at 80 degrees Celsius. Okay, at that time that sugar phosphate backbone will be bind tightly with the nitrocellulose membrane, and bases will be free. Okay, bases will be free. Here you can see these are the this is the nitrocellulose membrane, and these are the areas where that the single strand DNA is bound, phage DNA is bound here, and into this we'll add labeled probes, labeled radio labeled probes will be add onto this. Okay, radio labeled probes will be add this. And in the previous class and the that colony hybridization, I told that probes are the labeled oligonucleotides, and that probe will be highly specific to target DNA. Okay, so when we'll add that probe into this single stranded DNA containing nitrocellulose membrane, that probe will be bind onto this specific place DNA, and that then we will allow it for a hybridization, and we will wash this nitrocellulose membrane. That time the unbound probes will be removed from this, and the specific that probe will be bind on the specific area. Okay, that probe will be bind on the specific area, and after that we'll expose. Okay, this is marked, these probes are marked with the radio isotopes. That's why we'll expose this nitrocellulose membrane for to check the radio emission, that's radiation emission. So we'll check this nitrocellulose membrane for autoradiography. Okay, this for autoradiography we'll check this nitrocellulose membrane, and we'll expose into X-ray film. That time we can see one spot having, well, or this nitrocellulose the X-ray film will get a one spot. Sometime we'll get one spot, or sometime many spot will be get. Okay, on that depending on the spot, we'll compare this spot with our master plate. Okay, we'll compare with the master plate. Okay, this spot will be compared with our master plate. Here you can see this master plate will compare this spot of master plate, this spot with this master plate. Then we will isolate, we'll identify that which plaque having our recombinant phages. Okay, which comparing this spot observed in the autoradiography, we'll compare this with our master plate, and we'll identify which spot plaque having our recombinant phage. Okay, this is the plaque hybridization. I hope you understood. Okay. And the next technique is a immunological screening. Okay. Next technique is a immunological screening. Okay. In this technique, the desired recombinant clone can be isolated by detecting the protein or a polypeptide synthesized by a gene. Okay. In the previous techniques, we isolated the clones by detecting the sequence present in a recombinant DNA. But here, we'll detect the protein or peptide synthesized by a gene. Okay, that means in this technique, we'll isolate the desired recombinant DNA clone by detecting the protein or a polypeptide synthesized by a gene. Okay, in this technique, in that previous technique that we need that colony hybridization and the plaque hybridization, we need that DNA process. But here, instead of that, we need specific antibodies. Okay, specific antibodies we are required for the immunological screening of recombinant clones. Okay, this antibodies will be highly specific to proteins produced by this gene. Okay, protein produced by genes. Okay, that means this antibodies will be monoclonal and that will be specific to that proteins produced by the our desired gene. Okay, now we can see the steps involved in the immunological. Okay, now we have the transformed colonies. Okay, we having a transformed colonies. Okay, we having a transformed colonies in a plate. Okay, that we'll kept this plate as a master plate. Okay, this transformed containing plate will kept as a master plate, and we'll take this colonies by replica plating. Okay, we undergo for replica plating using that velvet mounted on a wooden block. Using that, we can make a replica plate into nitrocellulose membrane. Okay, we'll get a nitrocellulose exact copy with this colonies copy. We'll get in a nitrocellulose membrane, and we'll lyse this cell. Okay, we'll lyse this cells present on the nitrocellulose membrane. Okay, we'll lyse the colonies cells present on the nitrocellulose membrane, and will allow the proteins present in this colonies on this nitrocellulose membrane. Okay, allow this proteins to bind over this nitrocellulose membrane, and remaining part of the cells will be removed. That DNA or protein, DNA and RNA will be removed, or lipids will be removed. Only this colony containing proteins will be attached on this nitrocellulose membrane. Okay, this proteins colony containing proteins will be attached on this nitrocellulose membrane, and to that, okay, to this nitrocellulose protein containing nitrocellulose membrane will add primary antibody. Okay, we will add a primary antibody into this nitrocellulose membrane. Then that primary antibody will be highly specific to our the protein produced by our desired gene. Okay, protein produced by our desired gene. So this primary antibody will be bind over the this desired protein area. Okay, desired protein area or antibody will be bind. Then we'll add a secondary antibody. Okay, uh, okay, and after adding this primary antibody, we'll wash to remove the unbound antibody from this nitrocellulose membrane, and after the washing, we'll add a secondary antibody. Okay, we'll add a secondary antibody, and this secondary antibody will be specific to specific to our specific to primary antibody. Okay, this antibody is specific to our primary antibody, and the secondary antibody will be labeled. Okay, secondary antibody will be labeled. When this secondary antibody will add to this nitrocellulose membrane, this secondary antibody will be bind with the primary antibody is present over here. Okay, this primary antibody present over here. So this secondary antibody, then we'll wash this nitrocellulose membrane. That time, this unwound secondary antibody will be removed. Then there will be only bound antibody is there. Secondary antibody is there. So this secondary antibody will bind here only, because primary antibodies bind here only. Okay, that's why the secondary antibody bind on this only, and the other remaining antibodies will be secondary antibodies will wash over here because there is no other primary antibody, and we'll wash. Okay, we'll wash and we'll check. Okay, here enzyme is there, and we'll add is this enzyme substrate. Okay, we'll add the enzyme substrate. That time this enzyme will produce the degrade this using that substrate. This enzyme produce color. Okay, this enzyme produce color. That's color indicate the positive colony. That's color indicate the positive colony. You can compare this spot here in the master plate. Here this is a spot. Okay, that means this colony containing the desired our genes protein, desired genes protein. So we can identify that this is the desired desired colony. This contain our transgenic or that or recombinant DNA. Okay, this means in this the technique, first we have a master plate that's containing a transdermals, and this colonies will be transferred into nitrocellulose membrane, and we will lyse this cells of this nitrocellulose membrane, and will only this DNA this protein will be bound on this membrane, and into this that membrane will add primary antibody. That primary antibody will be highly specific to our desired uh, genes protein. Okay, how this our desired genes protein, and this primary antibody will be bind with the protein that's desired genes protein present in the nitrocellulose membrane, and the remaining primary antibody will be detached from here, and then we'll add a secondary antibody. Okay, this secondary antibody will be highly specific to the primary antibody. Okay, highly specific to the primary antibody. The primary antibody is only present here because this is the only uh, protein that is specific to this primary antibody. That's why this primary antibody is sorry, the secondary antibody is specific to primary antibody. Then this secondary antibody will be bind on this primary antibody. Okay, because secondary antibody is specific to primary antibody, that's why the secondary antibody will be bind on the primary antibody attached on a specific protein. And this secondary antibody, okay, secondary antibodies are the labeled antibodies. Okay, secondary antibodies are labeled antibodies. When we add substrate into this nitrocellulose membrane, that time this enzyme will convert, use that substrate, that will produce a blue colored, okay, that produce a that produce a colored compound. Okay, that will produce a colored compound. On detecting the color formation area, we can isolate which colony. Okay, here you can see, the primary antibody is attached on into that secondary antibody is also attached here. Here there is no specific antibody, the specific DNA protein is there. That's why there is no attachment of primary antibodies here, and that's why here also there is no secondary antibodies attached here, only secondary antibody attached here because primary antibody is there. And this enzyme will produce colored compound when added substrate, and here this location we are having the colored colony. Okay, will colored area. So this location we can compare with the our mass plate, we can identify which colony having the our desired transformed our desired recombinant DNA is present. Okay, that means in this mechanism we'll isolate the recombinant by depending on the protein synthesized by our desired DNA. Okay, this is the immunological screening of recombinants. I hope you understood this class. And the next technique is a screening by polymerase chain reaction. Okay, the next technique is a screening by polymerase chain reaction. That means PCR. Okay, polymerase chain reaction. Okay, in this technique, the desired clone can be isolated by amplifying specific genes. Okay, here we'll amplify the specific gene. Our desired gene will be amplified in this technique. Okay, in this procedure we have to uh, check by PCR. Okay, all colonies will be uh, checked by PCR to identify, to isolate the desired clone. Okay, it is possible to identify any clones by PCR only if there is an available information about the sequence to design suitable primers. Okay, to isolate the desired clone, we have to design the primers. Okay, to design the primers, we need some information about that specific sequence. Okay, if we having idea about the sequence of our desired DNA, then we can easily design the primers for the PCR, so we can easily select the desired clone from this mix of clones. Okay, for in this step with this PCR screening, first we have to this is the transformed plate. Okay, this is a transformed plate. We have to take individual colonies. Okay, we have to take individual colonies, and we have to, using our specific primers. Okay, using our specific primers against our target DNA. Okay, against our target DNA, we'll amplify individual colonies separately. Okay, we will amplify this individual colonies separately, and we'll undergo for agarose electrophoresis. Okay, we will undergo for agarose electrophoresis. And we'll check which well having amplification. If suppose this is the first colony and this is the first well, so here you can see the amplification, so we can identify it is a this colony is a containing our desired product. And this is the second well, third, fourth and fifth. Okay, if this is the second and this is the fourth and uh, this is the fifth. Okay, so you can see here five or six well containing this amplification. So you can see this is the first well, the second, third, fourth, fifth and sixth. Okay, sixth, but seventh, eighth and ninth there is no amplification. So this corresponding this first well, if it is a this is the first well sample, and this is the second and third, fourth and fifth and sixth. Okay, that's why we can say that this six colony have the our desired product. Okay, this six colony having our desired product, because this first six well containing the amplification. Okay, so the seventh, eighth, ninth well does not having any amplification. That's why the eighth, ninth, these are the not a desired recombinant. So this is the method used for the identification of recombinant clones using PCR. Okay, this is the identification of recombinant clones by PCR. Okay, I hope you understood this. So these are the methods, technique used for the selection and screening of recombinant clones. And if you have any questions, any doubt, please mark in your comment box. And if I found any corrections in this video, I will write in the description section of this video. So thank you for listening my class. Thank you.